Okay. Now, just so we know also, feel free to go ahead and ask any questions verbally as well. So you can do that, or you can type a question into that poll everywhere, and that will be fine. Let me go ahead and I'm going to turn off my video. There we go. Can someone just real quickly, so I know that I can actually hear you, because I've had malfunctions with this. Okay. Okay, great. Thank you. That's great. Okay, so all right, so I'm going to start going through these, and we'll work through this stuff. Okay, so it says, let's see here. All right, could we cover again brown adipose tissues and uncoupling proteins? Absolutely, that's a great place to start, and then I'll start to come back to this. So let me pull up here. Okay, so uncoupling is a major topic that we actually talked about within the lecture 9. And of course, we talked about like electron transport chain inhibition and ATP synthase inhibition and all sorts of things, okay? And uncoupling kind of falls within that. And there are unnatural, and then there are natural ways to uncouple these processes, okay? And when we say uncouple, what we mean by that is that without an uncoupler present, ATP synthase and the electron transport chain are codependent, meaning that they will not function unless there's a functional other portion of them. Okay, so let me go ahead and draw this up. All right, so we have something to kind of visualize here. And I'll use sort of something that you're used to seeing now, the way I diagram this in my excellent artistic ability. Okay, so here's ATP synthase. And when we say that these things are coupled, okay, we mean that this portion of it here, this would be the electron transport chain, that can't run without this portion, ATP synthase, okay, nor can ATP synthase run without the electron transport chain. They're codependent, okay? So that's what we mean by coupled, okay? Now, when we then talk about an uncoupler, okay, and the question that was asked was about brown adipose tissue, which means that there'd be natural uncoupling. So what I'm going to do here, I'm going to put an uncoupler over here, and we'll just make it a green box, okay? And this would be a protein that would be found expressed in mitochondria, in particular in brown adipose tissue, okay? And so we'll just call this UCP, uncoupling protein, okay? And what happens here is this, you have over here, we'll just draw NADH. NADH can be converted to NAD+, okay? And then those electrons will move, as you know, right, through these whole processes until you finally reach oxygen, making water. Oops. Okay. All right, and then we know this, that protons will be pumped, and I'm just going to put the number of protons here, will be pumped 4 at a time through that complex. They'll be pumped 4 at a time through this complex. Okay, and we're just ignoring mechanisms here, but that's what will happen. And 2 protons will be pumped through here. Okay, so all the redox reactions that will be encompassed in moving electrons from NADH to oxygen making water will result in a total of 10 protons moving from N to P side. Okay, so let me go ahead and put this N and P. Okay, all right, now when we start to then consider uncoupling an ATP synthase, we know that many of these protons will go through here, right, in this complicated process that we've talked about, and that will then generate or generate molecular force, okay, that can allow then for conformational changes and the production of ATP, except for I'm drawing this backwards. Let me do it right. Oops. ADP plus inorganic phosphate makes ATP. Okay? All right, so these protons here, 4 protons, 4 protons, 2 protons, okay? Those are going to then accumulate and they'll start to move through this, okay? And that will make ATP. Now, here's the interesting thing, okay, about brown adipose tissue. Brown adipose tissue will come equipped with this uncoupling protein that will then allow for these protons here to also move through this environment, okay? And they end up then on the inside, right? So instead of making ATP, they will make they'll do something else. They'll actually make heat, okay? And so we could just put it this way: the energy is going to come out of this as heat. Okay, when you start to move something, or in this case, the ions with their gradient, there's energy stored in that, and that energy is stored from the redox reactions of electrons moving from NADH to oxygen, right? That can be used to make ATP, but it can also, if it's not used to make ATP through this uncoupling proteins, it will actually generate heat. And so by and large, that actually will serve a purpose in brown adipose tissue. Okay, brown adipose tissue are found primarily in infants, and the adipose tissue found primarily in infants is brown. What we call brown adipose tissue is surrounding the vital organs. Okay, now the reason it's brown is because it's adipose tissue that has lots and lots of mitochondria in it. Okay, and that then gives the adipose tissue the kind of this brown tint to it. If you were to look at you and I's adipose tissue, okay, in our bodies, it would be primarily white. It doesn't have a ton of mitochondria in it, but it has a lot of lipid in it, and that lipid has more of a white tone to it, whereas the mitochondria give it more of a brown tone, right? But in any case, this brown adipose tissue, it has, of course, lipid content, but then it has all these mitochondria in it, and these mitochondria, each one of them comes equipped with multiple uncoupling proteins embedded in the inner membrane. Okay, and so what happens then, and the purpose of that, by the way, is that they can generate heat because the infant has a large surface to volume surface area to volume ratio, and they have a very difficult time regulating internal temperature, and that can be real dangerous for the vital organs. All right, so then these brown adipose tissue will actually generate this heat surrounding those organs to ensure that proper temperature is maintained, so that enzymatic catalysis and rates of enzymatic catalysis will occur at the proper rate, and there won't be tissue damage. All right, in particular in the vital organs, right? And we talked a little bit about the fact that can actually happen due to signaling. Norepinephrine can actually be released, I believe it's by the pituitary gland that recognizes cold temperatures, colder internal temperatures. And norepinephrine then will bind to these brown adipose tissue, actually at a beta adrenergic receptor that will then activate your and I's favorite protein from lecture 6, PKA, and PKA will activate fatty acid breakdown, what's called beta oxidation. We'll get to that later on. But then when fatty acids are starting to be broken down, the citric acid cycle rate will go way up, and they'll produce a lot of NADH, and then that'll produce a lot of protons on this inner membrane side, or inner membrane space side, that will then move through the uncoupling protein that generate the heat needed to maintain tissue integrity in those vital organs. So, okay, so hopefully that helps out. It's really, actually, I think, really fascinating when you look at it. So, okay. Got some other questions here, so let's look at this. All right, so free response questions 2, and 3. By the way, if someone says something on the chat, let me know. I'm not really not paying attention to the chat right now. I'm just going to kind of pay attention to this, but I guess I can look. Okay. All right, great. So there we go. So let's do this. Free response question 2 and 3. I didn't pull that up, but I will right now. So let me pull up the practice exam. If I can find them. Yeah, there we go. Practice exam 3. Actually, that's not the one I want to pull up. I'm surprised. I need to pull it up from here. Just a second. There we go. Okay, I was in the wrong folder. Okay, great. So practice exam 3, short answers. Okay, yes. Let's look at these. Okay, so let's look at number 1 here. Citric acid cycle is often referred to as the hub of metabolism as being an oxygen-dependent cycle. Predict how many ATP equivalents this cycle produces from the complete oxidation of acetyl-CoA to CO2 when combined with the processes of oxidative phosphorylation. How many ATP equivalents would this amount to if 1 molecule of glucose were the source of acetyl-CoA? Finally, describe the series of events that lead to inhibition of the citric acid cycle when oxygen is removed from the matrix of the mitochondria. All right, boy, this is a mouthful. What would this do to the rate of glycolysis and lactate production? Why? Okay. All right. So ATP equivalents. Let's talk about that first. This question, you would probably be right to ask yourself, okay, well, doesn't that depend on how many subunits are in my C ring? Okay. And so if you run into a question like this, where it's a short answer question where it doesn't have that on the exam, a C subunit of C ring of 9 subunits. Okay, and that will give an NADH-PO ratio. Will produce 2.5 ATP. Okay, and FADH2 or succinate will produce 1.5. Okay. Now, having said that, I don't think that there is a question on this next exam that it doesn't explicitly state C subunit or the amount of subunits in the C ring. Okay, but if you run into that, then that's what you want to do. Okay. Now it says this. It says right. The question is about the citric acid cycle. Predict how many ATP equivalents this cycle produces from the complete oxidation of acetyl-CoA to CO2. Okay. So when you do that, you have to just remember this, okay? And you just can't leave the citric acid cycle. I know that was on our last exam, but you can't leave it in this exam. It's just so intimately connected to oxidative phosphorylation, okay? But in that cycle, there are going to be 3 NADH made. Okay, actually, does the question? Oh, free response question 2 and 3. Sorry. Free response question 1 on practice exam. Okay, great. So we're going to do all 3 of them. People want help on all 3 of them. Okay, great. All right, so 3 NADH per cycle. Okay, and then really what we can just say 1 FADH2 per cycle, per turn of the cycle, and then 1 direct equivalent of ATP. I'll just put ATP. Is GTP or ATP depending on your tissue type. Okay, now this is going to then lead to, because we're multiply that 3 times 2.5, that's going to lead to 7.5 ATP per cycle, 1.5 ATP per cycle, and then of course just 1 ATP. So when we look at the cycle itself, right, you get what, 8, 9, 10 ATP per cycle. And that would be acetyl-CoA feeding into the cycle, right, at citrate synthase, combining with oxaloacetate, becoming citrate, and moving all the way around. NADH is produced by isocitrate dehydrogenase, is produced by alpha-ketoglutarate dehydrogenase complex, and also then finally it's produced by malate dehydrogenase. FAD is produced, or FADH2 is produced by succinate dehydrogenase, and then the ATP is by succinyl-CoA synthetase. Okay, all right, so that then, when you look up here, predict how many ATP equivalents this cycle produces for one complete oxidation rate. Okay, so we've done that, check mark. How many ATP equivalents would this amount to if one molecule of glucose were the source of acetyl-CoA? Okay, so what this is saying is, right, if we were to connect this to carbohydrate metabolism, how many would this lead to? And the answer to that would be to take this times 2, okay? Because with glucose, we produce 2 acetyl-CoAs, okay? So this would be then equal to 20 ATP per cycle, okay? And then finally describe the series of events that lead to the inhibition of the citric acid cycle when oxygen is removed from the matrix of the mitochondria. What would this do to the rate of glycolysis and lactate production? Okay, so let me go back to my last diagram. Right here? No, right here. There we go. All right, so if we come back here, the question is, what was the question again? Anaerobic conditions, I believe. But I want to just make sure. Describe the series of events that lead to the inhibition of the citric acid cycle when oxygen is removed, right? Anaerobic conditions. Okay, so when we come back here, all right, if you remove this, boom, you go anaerobic, okay, that will feed back that every little process that we talked about with all these complexes will just start to back up. They won't be able to move electrons any longer, okay? And so this whole thing will adopt the reduced state. That will cause NADH to increase in concentrations. Okay, this is very similar to what we talked about with our, I guess it was worksheet number 7. But in that case, we weren't anaerobic conditions, we were aerobic conditions, but we had cyanide that was doing this. Okay, but then NADH, what it's going to do, okay, is that it's going to inhibit the citric acid cycle. Okay, so if we come over here, pyruvate is converted to acetyl-CoA by the PDC, okay, and then acetyl-CoA feeds into this cycle, oxaloacetate, and acetyl-CoA will combine to make citrate. Okay, and then there will be a bunch of stuff that happens, okay, but I'll connect it to this. Succinate will be converted to fumarate. Okay, so there's several steps in here that I'm kind of just breezing over, just for brevity's sake, okay? But when NADH levels accumulate, they will feed back and they'll inhibit several steps in the citric acid cycle. They will also inhibit PDC. Okay, so PDC will be inhibited by NADH levels. Okay, and that will then prevent pyruvate from being converted to acetyl-CoA. Now, I won't draw it here just because it becomes complicated, but then what happens then in the cytosol, all right, is because pyruvate is not being converted to acetyl-CoA, it will be shunted, okay, or converted to lactate, all right, and lactate then will be excreted from these cells, all right, and that's an important feedback that we've talked about quite a bit, because it really does show the connections between oxidative phosphorylation, the citric acid cycle, and then finally glycolysis metabolism in the cytosol. So you'll definitely want to understand that. Okay. Okay. Questions on that? Feel free to mic in if you've got questions on that. No? Okay. All right, free response questions 2 and 3. So let's go there. Look at those. Oh, this one's, yeah, okay. So shown below is data derived from a suspension in a reaction vessel with buffer of mitochondria that have attained equilibrium and thus did not have an established proton gradient nor the substrates required to make ATP, which are ADP and inorganic phosphate, or to run the electron transport chain, which is succinate, at the beginning of the experiment. The mitochondria are intact and have functional enzymatic activity as well as transporters in their membrane. To generate these data, researchers added at the specific time points shown by arrows various compounds to the suspensions and then monitored oxygen consumption and ATP production. The bottom line represents APT production and the top line represents oxygen consumption. Oligomycin and like vintricidine, I honestly, I doubt that that's how you pronounce it, but are ATP synthase inhibitors. Given this information, answer the following questions. Okay, continue to the next page. Oh gosh. Okay, so what is this saying? So what this is saying is this: if you imagine a scientist or a group of scientists taking a whole bunch of mammalian cells or you know, eukaryotic cells, doesn't really matter, and breaking them open and then isolating from those cells mitochondria. So you can actually break open cells but leave mitochondria intact. And then there are various laboratory techniques that will allow us to isolate mitochondria from all the other cellular stuff. Okay. You can then, through a series of washes, essentially buffer washes, you can bring those mitochondria to equilibrium, meaning there's nothing really going on in them. Okay. But they're fully intact, meaning that they can carry out the processes of cellular respiration if they're given the right stuff. Right now, the right stuff is this. You have to have ADP and inorganic phosphate, and you have to have succinate. Okay, succinate is something that will kickstart the electron transport chain, and ADP and inorganic phosphate is the substrates needed to run ATP synthase. Okay, now because they're coupled, if you don't give them, well, if you don't give them the mitochondria sitting in the suspension, both of those things, then and oxygen, you have to bubble oxygen through this, then you can't get anything to happen, okay, because both processes rely on each other. In fact, if you look at this, come down here, right, the first thing that they add is succinate, okay? And so they add succinate, and you notice that the top line doesn't really change all that much, and that is an indication that oxygen is not being consumed, okay? And if oxygen is not being consumed, that means the electron transport chain is not running. Now, the bottom line represents ATP being made, okay? And if you're not consuming oxygen at the addition of succinate, then you're not making a proton gradient because electron transport chain is not running, and therefore you're not making ATP, okay? Now, the next addition, okay, this one, add ADP and inorganic phosphate. Well, now you've given everything that the mitochondria need. They have something to run the electron transport chain. They have something to run ATP synthase, all right? And so then what happens is that you see, first of all, oxygen consumption. That will make a proton gradient that can then be used to make ATP. So both lines go up because ATP is being made and oxygen is being consumed, okay? Now, they then go on and make this addition, oligomycin. Let's just talk about oligomycin. That's what we talked about in our class, right? That will inhibit ATP synthase. Well, if you inhibit ATP synthase, then because these two things are coupled, then obviously ATP synthesis will stop, but also at the same time, electron transport chain will stop because it's coupled to ATP synthesis, right? And then you finally add this thing. They finally add this thing called DMP, which is an uncoupler, and you can see what we mean by uncoupling. Oxygen is consumed in the absence of ATP being made. Okay, so the two processes are now quote unquote uncoupled. Okay, so that's the experiment. Okay, so when we look at this, then it says, given the chemiosmotic model, briefly explain why the initial addition of succinate does not lead to as large a change in the rate of oxygen consumption compared to the rate of change once both succinate and ADP and inorganic phosphate are present. Okay, so I have already explained that. Essentially, you have to have both processes, electron transport chain and ATP synthase, running to do this. There are a couple processes. Now, let me be a little bit more technical here. The reason you have to have ATP synthase running is because it maintains the proton gradient in a narrow concentration, if you will, or gradient. Okay, there's only so much energy found in an electron transport chain to continue to move protons across the inner membrane of the mitochondria. Well, if you don't have anything moving protons back from the p side into the inside, then the gradient becomes too steep, becomes too much energy for the electron transport chain to keep moving protons across the membrane. Well, ATP synthase is one of the molecules that does that. It allows protons to continue to move back into the inside so that the gradient doesn't become so steep that there's not enough energy in an electron transport chain to keep pumping protons. Okay, so when they're coupled, you got to have ATP synthase there to essentially regulate the gradient so that it doesn't get too steep. Okay, right now, oh yeah, here we go. So it says below, I would like you to draw the result of a similar experiment on the axis provided. However, you need to change the order of addition to be the following. Okay, see below. The solid line shown prior to line 1 represents oxygen consumption, and the dashed line ATP production. Please continue these lines starting at 1. Okay, so what this is saying is this. We are going to add, first of all, succinate. That's our first addition to mitochondria. So we reset the experiment, and when we reset the experiment, then we have those mitochondria in suspension, and the very first thing we do is add succinate, just like we did up here, okay? So you would expect the same thing to happen, right? You still have coupled processes, and so you don't expect much to happen, right? So I'm going to use this. Use black, like that. Not much is going to happen, and then I'll use a color, right? Dot 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 dot. That's going to not much is going to happen with ATP synthesis, right? So this is ATP synthesis. This is oxygen consumption. Okay. Now the next thing, interestingly, is DMP. So you uncouple them right away. Okay. And so when you uncouple them, then what's going to happen? Okay, is this. That oxygen is going to start to be consumed, even though ATP synthesis isn't going to really happen, okay, because there's nothing to make ATP, because ADP and inorganic phosphate aren't there. So ATP can't be made, but you can start to consume oxygen, okay? Now, after that, then we add oligomycin. Oligomycin then, right, is going to be added right here. And that then inhibits ATP synthase, but it doesn't do anything to electron transport chain because they're already uncoupled. So this is just going to continue to go up while this just flatlines, okay? Add ADP and inorganic phosphate, not much is going to happen here. You've already inhibited ATP synthase. You've already uncoupled the two processes. So this goes up, okay? And this just continues to flatline. And then finally, the last addition here is cyanide. And when you add cyanide, you inhibit the electron transport chain, and of course, ATP synthesis continues to flatline. Okay, so that's what's happening here in each of these additions, right? Once you add DMP there, the electron transport chain will just run as if everything is fully functional, okay, with ATP synthase, even though it's not, okay, because DMP is doing what that uncoupling protein, let's come back here, DMP is doing essentially this, okay? DMP, however, is an unnatural uncoupler, right? So the uncoupling protein is a natural one found in brown adipose tissue, whereas DMP is something that you have to ingest, and it's dangerous. It can be very dangerous stuff, so, okay. All right. I believe that answers question number 2 there. Let's go back here and look at this. I think that's it. Oh, yeah.
I have a question related to that.
Yeah, absolutely. What's up?
So if you didn't have the, the oligomycin or the ATP synthase inhibitor in there,
yeah,
and instead add a ADP and PI, would you see a small increase in ATP synthesis or
yeah,
not?
Yeah, great question. So the question that you're posing is this: what if we switch these, right? Or just removed oligomyosin altogether? What would that look like? Okay, so in that case, and your question is, would you see some ATP synthesized? And I think that you can make a solid argument for yes, that would go up. Okay. Um, it depends on how much DMP is there, right? So if you just loaded it up with DMP, then the likelihood of a lot of ATP being made from ATP synthase would be small. Okay. However, you could make a very solid argument that if your DMP concentrations are low enough, you can generate a strong enough proton gradient that ATP synthase, because it has the material needed to make ATP, could make some ATP. It wouldn't be as much as if DMP wasn't there, right? If DMP wasn't there, then it'd make more ATP. Okay, but it certainly could. Okay, good question. Other questions?
Thank you.
You're welcome. Other questions from anyone else? No? Okay. Well, let's keep going here. I'll try and get through as many questions as possible. Oh my goodness, here we go. All right, so describe the structural features of F1-FO ATP synthase and the mechanism by which it utilizes the energy of a proton gradient to drive the synthesis of 3 ATP molecules. After doing so, predict the number of protons needed to produce a single ATP molecule for a C subunit consisting of 9, 12, and 15 subunits. Okay, in your prediction, ensure that you include all protons needed to make ATP. Finally, how might this number change if DMP was infused into the mitochondria? Would it go up or down? Okay, and why? All right, so the question, this is a long question. There's a lot there. In particular, the first part of the question, I wrote this question with a practice question in mind, okay, or practice exam in mind, making sure that you do understand the structural features of F1-FO ATP synthase, right? So the whole premise of F1-FO ATP synthase is that you've got, and I'm going to butcher this, but I'll try and draw this out. Let's see here. We've got this C ring, right? Okay, so let's say that that's a C ring, okay? And then just to the side of that would be this C subunit, okay? The C subunit has these little channels. Not the C subunit, I'm sorry. The A subunit, okay? And it's got these channels that look something like this. It's so bad, okay? But you get the idea. Oops. So this would be the A subunit. Oh, that was the wrong thing to do. Oh well. Okay, right. So then, so what you have here is essentially that's the A subunit, and then connected to all this, okay, would be the central stalk. Okay, and then that would stick into the popsicle stick, if you will. Okay, and so then we'll just draw this. Okay, and gosh, it's really hard because I would have to draw all of them. All right, but you get the idea. And then there's this one here, and then there's one behind it. Okay, right. But anyway, that's the alpha and beta dimers. Okay, but the idea would be that you would have then here, so let's call this the P side here, and we would call this the N side here. And the idea would be then that you would have protons, let's say yellow dots are protons that would come into here, okay, and then they would leave out here. All right, and when they come in from this side, so this equals a proton. Right, when they come in from this side, then they cause this conformational change at this aspartate or this glutamate, right? Because they bind to this aspartate or glutamate, and that then causes this thing to start to shift in the what would be the clockwise direction, okay? And then there's one that's deprotonated, and so we have this proton flow. Well, that causes that rotation of the C ring causes this gamma shaft to spin, and then the alpha and beta dimers will start to change conformation, and that that central stalk will force a conformational change from the T state of an alpha and beta dimer to the O state. Okay, so T meaning ATP bound to the O state meaning open, and ATP will be knocked out. And that will happen for every 120-degree turn of the gamma shaft. Okay, all right, so so that's the structural features, okay? And then that's, you know, I mean, that's like 3 days worth of lecture combined into like, what, 5 minutes, okay? So obviously, you know, it's missing some detail. But generally speaking, that's what we're talking about. Now, when you look at this, then it says, well, you know, 9, 12, and 15. C ring consisting of 9, 12, and 15 C7. So I'm not going to do all of those. I'll let you guys practice some of the other ones. But let me go ahead and just do 12. Why not 12? Okay. And that's consistent with this last worksheet that we had. Okay. Well, 12 subunits means this. Okay. It means that 12 protons will be moved, okay, from the P side to the inside for a 360-degree turn of this C ring, okay? And a 360-degree turn equals the production of 3 ATP, okay? And this is, we can say this, if C ring is 12 subunits, it takes 12 protons, not 11. Not sure why I drew 11 there, but I did. 12 protons to give 360-degree turn. Okay. All right, so essentially what you have here then is this. You have some fairly straightforward and simple arithmetic to do, okay? 12 protons makes 3 ATP. So take 12 divided by 3, and that's then 4 protons per ATP. Now, don't you wish we were done? We're not done, okay? We have to add one more, okay? And that one more is to move inorganic phosphate in, right? So we have here ADP plus inorganic phosphate, and that will then make ATP. But you have to get this into the mitochondria, inorganic phosphate, and that takes 1 proton, okay, per ATP molecule. So we have to add plus 1 proton for PI. And that means that this equals 5 protons per ATP. Okay? All right. So it says this: after doing so, predict the number of protons needed to produce a single ATP molecule for a C subunit consisting of 9, 12, and 15 subunits. Okay, so there we go, 5 protons per ATP. All right, 4, 12. Okay, you can do, right, it's fairly simple arithmetic. You can start to move around these numbers and get different numbers. Okay, so I will say this: you want to understand why. Right, those numbers are the way they are, and I tried to explain that over here. Okay, all right. Now it goes on to say this: in your prediction, ensure that you include all protons. Okay, and then finally, how many and all protons, meaning this one, right? You have this extra one. Finally, how might this number change if DMP was infused into the mitochondria? Would it go up or down, and why? It would go up, most certainly. Okay, it would take more, quote unquote. I mean, it would still take this. That's kind of actually the way that question's worded is not quite right. It would actually be the same number of protons needed to make an ATP. Okay, and what I mean by that is the mechanism this doesn't change because we put in DMP into it. Okay, so it would still take a 360-degree turn, will still make 3 ATP. There's 12 subunits. It will still take 12 protons to do that. It will still take 4 protons per ATP, plus then the 1 proton to move the PI. So that would still be 5 protons. Now, what you might think, and this is why I said it would go up, you might think about the number of the actual PO ratios, right? Meaning this. you're going to have to pump more protons from the N to P side to get the 360-degree turn. Okay, because if there's let's say 5 of them that are needed to make ATP in this case, okay, and there's a 1-to-1 ratio without DMP for every proton that's pumped across the membrane with no uncoupler, that proton will then go through ATP synthase and make ATP. However, with the uncoupler, you could say that, well, one out of every 2 protons pumped from the N to P side will be used to make ATP. So you'll have to pump significantly more protons across the membrane to get the same number of ATP, right? Which means that you would have to increase consumption of energy in the form of like glucose or fatty acids. Okay, in fact, that's why DMP was this like wonder drug in terms of weight loss, because it would uncouple ATP synthase from the electron transport chain, and you'd have to consume more calories to maintain the same level of ATP or the steady state level of ATP in the cell. Okay, so that question, I'll be honest with you, that's not a great way to say it. Would it go up or down, and why? Well, it would be the same because the mechanism would be the same. But if it were to be worded as would you need more protons to be pumped from N to P side, would that number go up or down? And the number will go up. You'd have to consume more calories to produce more NADH, to produce more protons on the P side to keep the same number of ATP being produced. So, okay, hopefully that's not too confusing what I just said. Questions on that? No? Okay, let's see if we can't get through a couple more of these. So we're doing pretty good. Okay, so question 10, or question 3 from quiz 10. I have to pull up quiz 10. Just a second. I have to log into Canvas. And let's see here. Okay, let's see here. There we go. Quizzes. Quiz 10. Preview. Question number 3. Oh, more of this DMP stuff. So maybe I've already answered this question, but let's look. So the addition of 2,4-dinitrophenol, DMP, to a suspension of mitochondria in which an inhibitor of ATP synthase is present, along with a reduced electron carrier succinate, will cause which of the following? So that is really similar to question 2. Okay, in fact, it is, I mean, it's very similar to question 2 of our practice exam. So come back up here. So what it's saying is this. The infusion of, let me go back to this question. The infusion of DNT to a suspension of mitochondria in which the inhibitor of ATP synthase is present along with a reduced electron carrier succinate will cause which of the following? Okay, so essentially what we're saying is this. You would have your order of addition. Get rid of all this junk. Okay, your order of addition would be this. Okay, you have oligomyosin and then DMP. And ADP and intermediate phosphate will be up there. Okay, so you go succinate, let's say ADP and intermediate phosphate, and then finally you put oligomyosin in it. Okay, when you put oligomyosin into it, you would essentially stop ATP synthase, you would stop oxygen consumption. Okay, and then the question says, okay, well, what if I then put DMP in after I put in oligomyosin? Essentially this, actually. Okay, you have all this, you have oligomyosin, you add DMP. What's going to happen? Well, oxygen consumption is going to go up, ATP synthase will remain flatlined. Okay, so let's look at this. Decreased flux through complex 3. Okay, actually, there would be increased flux through complex 3 because all of a sudden you'd uncouple them. So that's not right. ATP synthesis would not happen because we've inhibited ATP synthase. Proton flow from N to P side via complex 2, certainly not. DNP does not induce all of a sudden magically complex 2 to move protons across the inner membrane. The concentration of NADH to increase in the matrix due to the subsequent inhibition of electron transport chain. Electron transport chain will not be inhibited. It will actually start to be activated because you've uncoupled them. Finally, then you're only left with increased rate of oxygen reduction, and that's right. So, okay. Great. Let's come here. Okay. Review of HIF1. Absolutely. Let's do that. So HIF1 stands for hypoxia-induced factor 1. We didn't talk about how it does things, but we talked about what it does, okay? So the how of it would really be a great topic for like molecular genetics, okay? So many of you will go on and take BC403 if you haven't already. Many of you have already taken something called Life 201, and you know that there are these then transcription factors that will increase rates of transcriptions of certain genes. Okay, and HIF1 is one of those. Okay, it will increase rate of transcription of certain genes. Right, and so what we have here, let me go to this. Let me get a blank sheet. Oops. All right, what we have here is this. You got a mitochondria. Okay, and then we've got our intermembrane space, and I'm just going to put the electron transport chain right here, ETC. And I'm just going to represent it as one big block, okay? And then, of course, we have ATP synthase here. And then we go like that, okay? Now, and you know, I don't want to spend too much time belaboring this point, but we know this, right? NADH goes to NAD+. And electrons flow through here, and then they're finally used to make water from oxygen. Okay, great. Okay, and then of course it makes proton gradient and ATP synthesis. Okay, now we also have then here, and this, you know, all these connections are super important. Okay, so we have pyruvate, and pyruvate is converted to acetyl-CoA, and that then feeds into citric acid cycle, oxoacetate, citrate, okay, and so on and so forth, and we know that this goes in that direction. Okay, there we go. All right, and we will also put, let's go ahead and I guess let's put the protons being moved through this and then back through this to make ATP. Okay, so that's everything that we talked about in lecture 9. Once again, diagrammed once again, okay? We also know that out here in the cytosol from our first portion of the class, that glucose is converted to pyruvate. Okay, small amount of ATP is made in that process. And a small amount of NAD plus is used up and NADH is made. Okay, great. So now, and then of course this flows through here. Okay, and there would have to be some transporters there, so we'll put a transporter there. Okay, there we go. So so that's, you know, the connection between all this stuff. Now, hypoxia-induced factor, okay, is something that the cell uses to recognize that oxygen levels are low, okay? And so if oxygen levels are low here, then this whole process in electron transport chain is going to slow down, okay, because we don't have that much oxygen. So these would be not quite anaerobic, but hypoxic, not a lot of oxygen there. And so what will happen then on a long-term scale is that can be harmful to the cell, because first of all, if you have low levels of oxygen, and you have large production of NADH, and you have a lot of electrons here, that oxygen can, when it's bored because there's nothing else to do, because well, okay, let's not use the bored terminology, but anyway, so the oxygen can actually snake in here and start to produce free radicals. Okay, not only that, but this is the major producer of ATP in the cell, right here, right? Everything down below in the mitochondria. You don't get a ton of it up here. Okay, so what happens is the cell will recognize that there's just not a lot of ATP production going on here, and there's potential for increased oxidative stress. Okay, lots of electrons, not very much oxygen to give it to, can make for oxygen-free radicals. Okay, and so what will happen is a hypoxia-induced factor is going to do this. It's going to, HIF1 will increase expression of GLUT transporters, in particular GLUT1. It will increase expression of glycolysis enzymes. And it will increase expression of PDC or PDH kinase. Okay. And so, and then also increase expression of LDH. So what you're going to do is this. You're going to get this massive increase in this, because all of a sudden you have more enzymes to carry out glycolysis. So this is going to go up. Okay. Not only that, but the amount of glucose coming into the cell because of this is going to go up. So these cells will start to absorb more glucose. They'll start to use more glucose to make energy. Okay. Now, at the same time, PDH kinase is going to inhibit PDC, okay, pyruvate dehydrogenase complex. And the reason is because then that prevents acetyl-CoA from being made, and that then slows this entire process down, the citric acid cycle. And that will slow down the production of NADH, which will slow down the production of electron transport chain, and that will then reduce the chances of oxidative stress. Okay, so once you have pyruvate PDH kinase, it will phosphorylate PDH, okay, and which is the first subunit of PDC, and that will then inhibit this whole process, and it will really slow everything down here. At the same time, you have this increased expression of LDH, so that pyruvate will be converted to lactate. Okay, so that this can happen. Right now, this is so what you have then in these cells that have large expression of HIF1, you'll have reduced capacity of electron transport chain, oxidative phosphorylation, citric acid cycle, but you have increased capacity of glycolysis and lactate production. So these cells will start to absorb lots of glucose, and they'll start to excrete lots of lactate. Well, that's the phenotype of most cancer cells. Okay, in fact, we find in many cancer cells a high level of HIF1. Okay, and that was recognized long ago, back in the 1920s, and it's called the Warburg effect. Okay, so anyway, that's why we spent some time talking about it. Okay, all right. Interesting questions, interesting stuff, fascinating. Okay. Questions on that? All right, so let's get through. Let me do one more question here, okay? Because I'm running out of time here. It's almost 4 o'clock. It says, what if you inhibited the Q cycle? Well, if you inhibited the Q cycle, so let's come back to our drawings here that we have. If you inhibit the Q cycle, essentially you're going to have the same thing as everything else. You just stop it here, okay, instead of stopping it at complex 4. We talked about cyanide, and cyanide will stop it at complex 4, right? And that will then lead to an entire, every single thing in the electron transport chain obtained in a reduced state. Okay, so if you stop it at complex 3, then what will happen is that cytochrome C, which feeds electrons to complex 4, as well as all the redox centers in complex 4, will actually obtain an oxidized state because electrons can't get to them. Okay, and then, but everything else past this point, okay, will be the same. NADH will accumulate. That will come back and inhibit the citric acid cycle, inhibit PDC. That will then lead to decreased production of acetyl-CoA from pyruvate. Decreased production of acetyl-CoA from pyruvate will mean increased production of lactate, and lactate will be excreted. Okay, all right, so similar things, okay? Okay, so let me go ahead and stop right there, all right? Unless there's any other, well, let me just stop right there. So, and let all of you go. I know that many of you have to go to class and everything. So, if you haven't put a remark in the chat, please do, so that I can take your attendance and you get credit for being here. And I will post this recording if you want to listen to it or something like that again tomorrow. So, okay. All right. You're welcome, everyone. Everyone's saying thank you. You're most welcome. If you have questions, additional questions, by all means, go ahead and email me. Okay. All right. Take care, everyone. Have a good rest of your day.
Thank you.
You're welcome.